So in our previous analysis, we always looked at snapshot data. So what does that mean? Once we measure a cell, we only have one measurement of the full transcriptome, and afterwards the cell is gone. So we cannot really learn anything about its fate or where it should have or would have gone to if we would have let it. The concept of RNA velocity is breaking that snapshot idea of single-cell RNA sequencing data, and what I depicted here is a UMAP of the dentine gyrus development. And what you see here are these flow errors that are now an inference of the cell fate from the transcriptome. And I tell you in a minute how this is actually possible. When we spoke about a gene structure and the transcripts earlier last week, I showed you that the gene is first of all structured in exons and introns. So exons are here these brown blocks, and the introns are the lines. When we capture RNAs, they come from the whole gene structure. In the UMI-based setting, they're predominantly from the last exon. However, sometimes these reads that we capture can span this exon-exon boundary, which means that this intron already has been cut out. So that means that these reads are already spliced and mature and ready to be exported into the cytoplasm and turned into proteins. On the other hand, we also observe unspliced reads, so short sequences that span the exon-intron boundary. That means these reads are immature and still have to be processed. And now the interesting part is that we can use these observation of spliced and unspliced read to learn something about the gene dynamics, so when a gene is turned on or turned off. In the different protocols, we observe different ratios of unspliced and spliced reads. For example, the full-length protocol SmartSeq contains almost a quarter of unspliced reads, while the 3' bias protocols, INDROP or Chromium, so 10x genomics pipeline, they only contain like 10% of unspliced reads. So how do these reads help us to learn something about gene dynamics? We can model the gene dynamic with simple mass action kinetics. So we have here a gene that is either where the transcription is either turned on or off with these rates alpha on and alpha off. And once gene expression is, or gene transcription is turned on, this gene is transcribed with this rate alpha into an unspliced mRNA that still contains both exons and introns. This unspliced immature mRNA undergoes splicing and other processing steps to make a spliced RNA where all the exons are concatenated together. This spliced RNA is then finally transported to the cytoplasm towards the ribosomes to produce proteins. At the end of an mRNA lifetime, it's degraded with this rate gamma. So we can summarize this process with a system of ordinary differential equations where u dot of t is the derivative of the unspliced mRNAs after the time derivative t, and we have the same for the spliced read after the time. So from this model, in order to solve it, we can derive a steady state model that has been done in this VeloCyto project that was published in Nature 2018 by Joele Lamano. And what they estimate now is when we say steady state is that we set these two ODE equations to 0. And then we can solve it. We set this beta parameter to 1 for identifiability reasons. And then we can derive that the ratio of unspliced reads equals this normalized gamma rate, so the degradation rate, times the number of spliced reads. Anything that deviates from the steady state is a so-called velocity. So we define a velocity of a gene of by the unspliced number of reads minus the degradation rate times the number of spliced reads. That's a lot of formulas, so let us fill this process with pictures and follow this kinetics. So what does steady state first of all mean? When a gene transcription is turned on, and we can follow it over time, the gene the number of spliced reads comes to a so-called plateau. So, and this is the steady state. So here the number of spliced reads does not change anymore until we have a turn off of the transcription. When we want to look at spliced and unspliced reads at the time, we use a so-called phase diagram. Here on the x-axis we have the spliced reads versus the unspliced reads on the y-axis. And we can immediately find these two processes of the transcription being turned on here on this green curve and transcription being turned off on the red curve. The upper end and the lower end of these two processes, where the red curve and the green curve meet, these are the steady states. So we have either a steady state where no transcription is taking place, or where we have an equality, a balance of spliced and unspliced reads. This linear line here has the slope gamma. And now the velocity of each gene is now the difference of the red or the green line towards this linear slope. So the further away a cell is on this phase diagram, the larger its gene velocity is. This model has a couple of drawbacks. First of all, we assume that we actually reach the steady state with gene expression. However, if you have a fast-going process, as depicted here, or fast on and off switch of gene expression or gene transcription, then you don't necessarily reach the plateau. So you don't reach the steady state, and therefore the steady state is not present in your data. But still the model assumes that we reach a steady state. So, and when we always assume we use a steady state, we don't use all of the cells to estimate the gene dynamics. We only use the cells from the steady state. Further, we do not, the model is not fully identifiable, so we have to fix this beta rate of splicing to a certain factor in order to make this model work. However, splicing rates can be different. They can also be different across genes, and this is something we don't account for in the steady-state model. So my colleague Volker developed a dynamical model to address all of these issues, and where he also uses all the cells to learn dynamics of the cells. So on the one hand, we have now the steady-state model. Now on the other hand, we have the dynamical model. I tell you in a minute what's the difference of these two models. So again, we have this model of transcription. So we have transcriptions either on or off with rate alpha. We have the splicing, we have the degradation. So using a dynamic model means that we use, first of all the rates. So we have parameters for the reaction rates. So we summarize them in this parameter theta. And furthermore, we introduce latent variables, which are not present in the ODE system. So we have at switching time, we have the time state, and we have a so-called, let's say a state parameter. I'll tell you in a minute how we assign different states. We can solve the ODE model, the system of ordinary differential equations in a linear algebra fashion, and now can do a parameter estimation for all the different parameters that we have here in the model. So a word on the states. We assign to every cell a certain state where it is in. Either it's in the so-called on state, or it is in the off state, or it is in the steady state. So these are the 3 different types of states a cell can be potentially in. We write this up as a likelihood model. So we determine the likelihood of the unspliced reads and the spliced reads with respect to our reaction rates and the latent variable. And again, the unspliced reads and the spliced reads are subject to the time, the state, and the reaction rates. So what we now have to estimate is the reaction rates and the latent variables. It looks complex, potentially it is, but we solve this with expectation maximization algorithms. So we compute the likelihood, optimize the parameters, so that's the maximization step, and update until we converge. So let's fill this with pictures. So first of all, we assign states to all of the cells, what is depicted here with the different colors. Then we compute a latent time so that we can assign the cells either to the on state or the off state. Then the cells are fitted towards this curve. And then we update the model parameters, which means that we in fact actually update the shape of the of this curve of upregulation and downregulation or off switching of the genes. This process is iterated over until the likelihood model is maximized, and then we return the parameters for the model. And yes, this model is optimized for every single gene. The advantage of this model is that we don't have to assign some cells to the upper steady state, but we can actually determine a hypothetical steady state, which is not necessarily in the data, but we use the dynamics of gene transcription being turned on and off to determine this hypothetical steady state. And with this Dynamical model, we can get a more accurate description of the gene dynamics. What you see here, we don't assign, so we some kind of a vector field that you see here, that the cells are aligned along this dynamics and not aligned by the slope, or just assigned to the slope that you would have had with the steady state model.